Dear students, I am Dr. Muhammad Safdar. I am working as an instructor of biology at Virtual University of Pakistan. I will teach you in this course Biosensors. In this course, we will cover the following contents such as what is biosensors, what are their components, what are their types, and their applications. In this module, I will teach you what is the biosensors and what are the definitions towards a concept of biosensor. A biosensor is a self-contained integrated device that is capable of providing specific quantitative or semi-quantitative analytical information using a biological recognition element which is in direct spectral contact with a transduction element. A biosensor is an analytical device which converts a biological response into an electrical signal. What is the terms of biosensor? The biosensor is often used to cover sensor devices which are used in order to determine the concentration of substances and other parameters of biological interest, even where they do not utilize a biological system directly. The emphasis of this module concerns enzymes as the biological responsive material, but it should be recognized that other biological systems may be utilized by biosensors. For example, whole cell metabolism, ligand binding, and the antibody-antigen reaction. Biosensors represent a rapidly expanding field at the present time, with an estimated 60% annual growth rate. The major contribution is coming from the healthcare industry, which is about 6% in the Western world, who are diabetic patients and would benefit from the availability of a rapid, accurate, and simple biosensors for glucose. But the people from the food quality and environmental monitorings are also using them. Biosensor, a device that uses a specific biochemical reactions to detect chemical compounds in biological samples. I'm going to explain you via an example, in which example we will monitor mutagens. How we can monitor a mutagen by a biosensor? Here is a blue laser light which will pass through the tissue from where it will be detected the mutagen and show the possible carcinogens in the form of electrical signal. Current definition: a sensor that integrates a biological element with physiochemical transducer to produce an electronic signal which proportional to a single analyte, which is then converted to a detector. Here I am going to give you some examples like potentiometric biosensors, amperometric biosensors, and colorimetric biosensors. Also, biosensors can be used for the detection of food compounds, study of biomolecules and their interactions, drug development, crime detection, medical diagnosis, and environmental field monitoring. Thank you.